Well, students, we'll now begin a chapter on the living cell, the smallest living thing, and we'll see that life is based on chemistry operating the cells. That is, living systems are chemical machines. But first, let's ask the more general question: what is life anyway? Now, usually when we see something that we think may be alive, we will poke at it or shine light on it, see if it responds, or see if it responds to heat or to sound. Think of all those sci-fi movies where they find something frozen in the ice and they chip away at it and they see if it moves. But what are the kind of things that we really look for to see if something is alive, to see if it is life? Well, we look for organization. Is it made of a cell or many cells? And we'll see that organization is really important for life. That is, you can take a living cell and put it into a blender and blend it all up, and it's no longer alive. It has to be structured in a particular way to be alive. We'll see that a very important characteristic of living systems is that they transduce energy transduction. That is, they must have some fuel, some source of energy to make, and we'll see this molecule called ATP, to make ATP, which is then used to perform or to drive various types of biological work. Now, those types of work could be such things as reproduction, because to be alive, you have to be able to make another copy of yourself. That it could be either asexual reproduction, where a cell just divides into two, or sexual reproduction, where you have a mother and a father as parents that combine their genetic information in different ways to make an offspring. And that'll get into topics of genetics and DNA. Also, energy is used to maintain, to regulate what we call homeostasis. That is, a cell will maintain its temperature to the extent it can, pH, the composition of the various ions inside of the cell, and to regulate the amount of various metabolites and fuel molecules inside of the cell. Also, a cell in larger organisms will use ATP to grow, a process we call anabolism. We already talked about responding to stimuli, that is, things that are such as chemotaxis, that is, responding to chemicals, phototropism, responding to light. Irritability, we say that cells are irritable, meaning that if we sort of poke at them in various ways, including chemical ways or electric ways, they will respond. And also, adaptation to the environment and evolution are characteristics of living systems. But let's begin by talking about some of the simplest cells and talk about their sizes and shapes. And I show on this slide three different major categories. Plant cells in the upper right, showing that these are fairly regular, almost square or regularly shaped. They stack together quite a bit. And we see in the left, we see bacteria. They just happen to be painted green in this slide. But bacteria, which have different shapes, from just spherical to long squiggly things, and oftentimes they have tails or flagella so they can kind of swim around. Sometimes they have spikes on their surface. And then we see human or animal cells on the right, and we see also that they come in a variety of shapes, from the fairly simple red blood cell to spherical cells to elongated cells such as muscle cells and nerve cells, and cells that have flagella like sperm cells. So lots of different shapes. Now, to give you a little bit of history, let's go back into like 1663 with Robert Hooke. Now, he would have been a contemporary or in between the times of Galileo. Galileo, of course, developed the telescope and also did some studies with microscopes. And Robert Hooke used some of these microscopes to study plant cells, including cork, and an image of cork cells is shown to the right. And that he was followed by Anton van Leeuwenhoek, the first person that we would have called a microbiologist, who took pond water and blood and saliva, semen and feces, and looked at these under the microscope to see living cells. And he called these cells animalcules. Now we have two kind of versions of microscopes. The first microscopes were dependent just on light and a human's ability to just sort of look through lenses and with his or her own eyes to see things, called light microscopes. We can amplify images to a certain extent. And a more modern development is what is called an electron microscope, which allows us to view even smaller subcellular organelles. In 1838, this German botanist named Matthias Schleiden was the first to propose what is called the cell theory. And that cell theory, drum roll, cell theory, is that all life forms are composed of cells. Okay, I think we've got you there on that one already. All life forms are composed of cells. And that the cell is the fundamental unit of life. That is, we are multi-organed and multi-celled life forms, but we're made of different types of cells working together. And a third tenet is that all cells arise from previous cells. Even we arose from a fertilized egg that developed into all of the different cells in our bodies. One important characteristic of a cell, and we're beginning talking about the structure of cells in the next slides and in the next video. One important characteristic is that there must be insides of a cell that are separated and protected from the environment, from the outsides. You have to have a way to separate the insides, the cell components, from the outsides, from the environment. And this involves either a cell membrane and or a cell wall. Now, when we'll first quickly talk about cell walls. These are found in bacteria and fungi and plants and algae. They're a more rigid structure made of polysaccharides such as cellulose and other polymers. And they provide some rigidity to the structure. And shown to the right is an example of a plant cell, where you can see a fairly regular, in this case like a pentagon, shaped external cell wall, and inside you'll see the cell. The blue stuff would be parts of the cell. So plants have cell walls, but they usually also have an internal cellular membrane, also called a plasma membrane, which is based on lipid components, what is called a lipid bilayer, where you have two layers, and if you look at the bottom right, those kind of orangey circular things are part of lipid molecules. They are most likely phospholipids, and they have a part, the circular part drawn here, which will interact with water. It's called the head group, and they will have long tails, shown as sort of gray tails in this drawing, which will not interact with water. So what happens is that the tails will sort of stick together because they're trying to avoid contact with water, and the polar head groups will interact with water on each side of what is called a bilayer membrane. So the bilayer membranes construct the complete plasma membrane around the cell, and also many subcellular organelles will be wrapped with or enclosed in some type of bilayer membrane. Now, these bilayer membranes are important because not only do they protect the inside from the outside components, but they also have selective permeability properties. What that means is that they will allow some things to go in and some things not to go in. That is, they're kind of like a boundary. And so there will be pores or channels or transport systems to allow certain nutrients, certain ions, certain other molecules to go from the outside to the inside, and vice versa, to be secreted from the inside of a cell to the outside. And some of these processes involve what we call receptor molecules. So a receptor molecule, which is usually a protein, will be embedded in a bilayer membrane, and it might recognize some molecule, such as a hormone, such as insulin, from the outside, and this external molecule, such as a hormone, might bind to this receptor, and that will trigger either bringing that molecule inside the cell or trigger some processes to occur inside of the cell. Now we're going to pause now, again, have a little quiz, and then we'll move on to our next set of lectures, which will be about subcellular organelles and other subcellular structures.